In the darkness of an ancient ocean, two cells drifted toward each other. They had no brain to recognize the moment, no memory to record what would happen next. But in the space between them, smaller than a grain of sand, wider than the gulf between stars, the entire future of life hung suspended. For 1 billion years, every cell that had ever lived had followed one rule: survive alone, die alone. Competition was the only language life knew how to speak. Until this moment, until these two cells did something that should have been impossible. They chose each other. What happened in that microscopic space would echo through time. It would build forests and fabricate mines. It would create love and construct civilizations. Because when life discovered teamwork, it discovered how to become more than the sum of its parts. It discovered how to become us. But here's what nobody tells you about cooperation. It began as a mistake, a failure so catastrophic that it should have meant death for both cells involved. Instead, it became the most successful accident in the history of existence. According to microbiologist Lynn Margulis, who spent her career studying these ancient alliances, the first act of cooperation was actually an attempted murder, one cell trying to consume another, both failing so spectacularly that they became something neither could have imagined. They became one. Tonight we're diving into the precise moment when life learned its most powerful secret, when the universe discovered that the only way to build complexity was to break the oldest rule of survival, when competition gave way to collaboration, and in doing so, wrote the first line of every love story, every friendship, every family that would ever exist. If you've ever wondered why humans form tribes, why your body works as one system instead of 37 trillion competing cells, why cooperation feels so right even in a world that rewards competition, the answer lives in this moment. The moment when two ancient cells looked at infinity alone and chose the terrifying alternative together. "3 billion years ago, Earth was an alien world. No flowers painted the landscape, no birds carved paths through the sky. No creatures crawled across the seafloor. There was only water, rock, and a thin soup of single cells drifting through endless darkness. And for 1 billion years that was enough. Picture the vastness of it. An entire planet wrapped in ocean, and in every drop of that ocean a war. Not a war of armies or ideologies, but something simpler and more brutal. Every cell for itself. Every organism a solitary empire of one, competing for the same dissolving minerals, the same fleeting photons of sunlight that managed to penetrate the murky depths. These weren't primitive beings, not in the way we might imagine. According to research from the NASA Astrobiology Institute, these early cells had already mastered the fundamental tricks of existence. They could harvest energy from chemicals, they could replicate their DNA with stunning accuracy, they could sense their environment and respond to it. Some could even swim, using whip-like flagella to navigate their liquid universe. They were in every sense that mattered perfect at being alone. The chemistry of these ancient cells was remarkably sophisticated. They possessed enzymatic machinery that would put modern factories to shame. Ribosomes, those protein-building machines, were already achieving accuracy rates of 99.99% in translating genetic code. DNA polymerases could copy billions of base pairs with fewer errors than a medieval scribe copying a single page. These cells had learned to build complex lipid membranes that could selectively allow certain molecules in while keeping others out. They had evolved protein pumps that could move materials against concentration gradients, defying entropy itself. Dr. William Martin from Heinrich Heine University describes these early cells as thermodynamic miracles. Each one was a tiny engine that could capture energy from its environment and use it to maintain order in a universe trending toward chaos. Some harvested light, splitting water molecules with molecular machinery so efficient that human technology still can't match it. Others oxidized sulfur compounds, extracting energy from rocks themselves. Still others had learned chemosynthesis, building organic molecules from inorganic carbon using nothing but chemical energy. But perfection, it turns out, has limits. The ocean was changing. Oxygen, that element we now depend on with every breath, was beginning to poison the water. Cyanobacteria, those blue-green architects of atmosphere, had learned to crack water molecules apart and harvest the hydrogen, discarding oxygen as waste. What they didn't know was that their waste would become the planet's first mass extinction event. The Great Oxidation Event, scientists call it now. Back then, it was simply the end of the world. The scale of this catastrophe is difficult to comprehend. Dr. Timothy Lyons from UC Riverside studies ancient ocean chemistry through rock formations that preserve chemical signatures from this period. His research reveals that oxygen levels rose from essentially 0 to about 2% of current levels over just a few million years. For organisms that had evolved in an oxygen-free world, this was like suddenly filling the atmosphere with chlorine gas. For the anaerobic cells that had ruled Earth for 2 billion years, oxygen was death itself. It ripped apart their proteins, shredded their DNA, turned their own chemistry against them. The oceans became killing fields. 99% of all life on Earth would die in this catastrophe. But in the depths of this disaster, something unprecedented was about to happen. The chemistry of oxygen toxicity is particularly brutal. Oxygen readily accepts electrons, forming reactive oxygen species like superoxide and hydrogen peroxide. These molecules are cellular wrecking balls, breaking carbon-carbon bonds, oxidizing amino acids, and causing DNA strand breaks. Modern cells have elaborate antioxidant systems to neutralize these threats. Catalase, superoxide dismutase, glutathione peroxidase. But ancient anaerobic cells had none of these defenses. They were naked before the oxygen storm. See, when you're drowning in poison, when the very air has turned against you, the ancient law of competition starts to look less like wisdom and more like suicide. Some cells found ways to hide from the oxygen, burrowing into mud, fleeing to the deepest trenches where the poison hadn't yet reached. But a few cells tried something different, something that had never been attempted in 2 billion years of evolution. They got close to each other, dangerously close. The scientist who first proposed this radical idea was ridiculed for years. Lynn Margulis, working at Boston University in the 1960s, suggested that the complex cells that make up all plants, animals and fungi arose from an ancient merger. Her paper was rejected 15 times before finally being published in 1967. The scientific community called it fantasy, impossible, a violation of everything they knew about evolution. The resistance to Margulis's idea wasn't just scientific conservatism, it challenged the fundamental narrative of evolution as a story of competition. Darwin himself had written about the struggle for existence. The phrase survival of the fittest had become synonymous with evolution in the public mind. Here was Margulis suggesting that the most important event in evolutionary history wasn't competition, but cooperation. It was heresy. But Margulis had seen something others had missed. She noticed that mitochondria, those power plants inside our cells, had their own DNA. They divided on their own schedule. They had double membranes, as if one cell had been wrapped inside another. They even had their own ribosomes, slightly different from the cell's own protein factories. These weren't just organs that had evolved inside cells, these were immigrants, ancient visitors who had never left. The evidence was overwhelming once people started looking. According to research from the University of California, the genetic code of mitochondria is more similar to certain bacteria than it is to the cells they live in. It's as if you opened up your laptop and found a completely different computer running inside it, speaking a different programming language, yet somehow making the whole system work better. The genetic evidence is particularly compelling. Mitochondrial DNA uses a slightly different genetic code than nuclear DNA. In mitochondria, the codon UGA codes for tryptophan instead of being a stop codon. The codon AGA is a stop signal instead of coding for arginine. These differences make no sense if mitochondria evolve from within eukaryotic cells, but they make perfect sense if mitochondria are descended from bacteria that were captured and domesticated. But how did that first encounter actually unfold? Dr. Nick Lane from University College London has spent his career reconstructing this moment. His models, published in Cell in 2023, suggest it began with hunger. A large, slow archaeon cell struggling in the oxygen-rich waters encountered a small, fast bacterium that had learned to process oxygen. The archaeon did what cells had done for billions of years. It attacked, it engulfed, it tried to consume. the mechanics of cellular predation are surprisingly complex. The archaeon would have extended pseudopods, finger-like projections of its membrane, to surround the bacterium. This process, called phagocytosis, requires sophisticated molecular machinery. Actin filaments must polymerize to push the membrane forward. Myosin motors must contract to pull the prey inside. Membrane fusion proteins must seal the compartment. It's a molecular ballet that modern cells still perform billions of times each day. Here's where physics becomes poetry. The bacterium should have been digested, should have been broken down into component parts, its proteins recycled, its DNA discarded. Instead, something extraordinary happened. The bacterium's ability to process oxygen created a gradient, a flow of energy that the archaeon had never experienced before. It was like a starving creature suddenly tasting sugar for the first time. The archaeon didn't digest its prey because the prey was more valuable alive. The energetics of this partnership were revolutionary. Before the merger, the archaeon might have generated 2 ATP molecules per glucose molecule through fermentation. The bacterium, with its oxygen processing abilities, could generate 36 ATP molecules from the same glucose. Together, they had an 18-fold increase in energy availability. It was like going from a candle to a power plant overnight. But the bacterium faced its own crisis. Trapped inside another cell, it should have suffocated, should have been cut off from nutrients. Instead, it found itself in a protected environment, surrounded by resources, shielded from the toxic oxygen levels outside. The prison became a paradise. The predator became a protector. Inside your cells right now, 37 trillion times over, an ancient negotiation continues. Every mitochondrion maintains its own genetic material, about 37 genes in humans, while the cell's nucleus holds the remaining 1,500 genes that once belonged to that original bacterial partner. It's a distributed system, like a business where some decisions are made in branch offices, while others require approval from headquarters. This division of genetic labour didn't happen randomly. The genes that remained in mitochondria are primarily those encoding proteins involved in the electron transport chain, the core of energy production. These proteins must be produced quickly in response to changing energy demands. Having their genes locally allows rapid response. It's like keeping emergency supplies in every room rather than in a central warehouse. Dr. Ian Johnston from the University of Bergen studies this cellular communication. His 2024 research reveals that mitochondria and their host cells engage in constant molecular dialogue. When the cell needs energy, it sends chemical signals to the mitochondria. The mitochondria respond not just by producing ATP, the universal energy currency, but by adjusting hundreds of cellular processes. They influence when cells divide, when they die, even when they decide to become cancer. The signaling between mitochondria and nucleus is bewilderingly complex. Calcium ions flow back and forth, creating waves of information. Reactive oxygen species, once thought to be purely harmful, act as signaling molecules at low concentrations. Metabolites like acetyl-CoA and NAD+ carry information about energy status. Small peptides encoded by mitochondrial DNA can even travel to the nucleus to influence gene expression. It's a conversation that never stops, a dialogue that has been refined over 2 billion years. This isn't just cooperation, it's integration so complete that neither partner can survive alone. Scientists have tried to remove mitochondria from cells to see if modern cells can live without their ancient partners. The cells die within hours. Conversely, mitochondria extracted from cells and given all the nutrients they could want still perish quickly. The partnership that began as optional became mandatory. The merger that started as temporary became permanent. The intimacy of this relationship extends to the molecular level. Mitochondrial proteins made in the cytoplasm must be tagged with specific targeting sequences, molecular zip codes that ensure they reach their destination. These sequences are recognized by receptors on the mitochondrial surface. TOM20, TOM22, TOM70. Each receptor recognizes different targeting sequences. It's a postal system more complex than anything humans have designed. But how did this molecular marriage actually work? The mechanics are more intricate than any human technology. According to research from the Max Planck Institute for Biology, the process involves 5 distinct systems working in harmony. First, the protein import machinery. Mitochondria can't make all their own proteins anymore. They depend on proteins made in the cell's cytoplasm, which must then be threaded through specialized pores in the mitochondrial membranes. The complexity of protein import defies belief. Proteins must be unfolded to pass through the import channels, then refolded on the other side. This requires molecular chaperones, proteins whose sole job is helping other proteins fold correctly. HSP70 in the cytoplasm keeps proteins unfolded. MT, HSP70 in the mitochondrial matrix pulls them through. HSP60 and HSP10 form barrel-shaped chambers where proteins can fold in isolation, protected from the cellular environment. The partnership between that first archaeon and bacterium was just the beginning. Once life discovered that cooperation could overcome competition, the strategy spread like wildfire through evolution's playbook. But not in the way you might expect. It wasn't a gradual transition. It was an explosion. Between the first eukaryotic cells and the emergence of true multicellularity, there was a period scientists call the boring billion, a billion years when not much seemed to happen. But beneath the surface, cells were experimenting with different ways of being together. Colonies formed and dissolved. Cells stuck together temporarily, then separated. It was like evolution was learning the grammar of cooperation before writing its masterpiece. Dr. Rosie Allegado from the University of Hawaii studies these transitional forms. Her work on choanoflagellates reveals that the switch between single-cell and colonial living can be triggered by molecules from bacteria. A single bacterial protein can cause choanoflagellates to form rosette-shaped colonies. It's cooperation induced by communication between different domains of life. Bacteria talking to proto-animals in chemical languages that predate words by billions of years. For nearly 2 billion years after that first merger, life remained mostly microscopic. Complex cells with mitochondria existed, but they seemed content to remain single. Then, around 600 million years ago, something shifted. The fossil record from this period, particularly the Ediacaran biota discovered in Australia, shows something unprecedented. Cells weren't just tolerating each other, they were organising, they were specialising, they were becoming something that had never existed before. Multicellular organisms. The Ediacaran fossils are unlike anything alive today. Dickinsonia, looking like a quilted bath mat, could grow over a metre long. Tribrachidium had threefold symmetry, unlike the bilateral or radial symmetry of modern animals. Charnia looked like a fern but was actually an animal, feeding by absorbing nutrients through its surface. These were evolution's rough drafts, experiments in multicellular living that would mostly end in extinction. Dr. Nicole King from UC Berkeley has spent her career studying this transition. Her research on choanoflagellates, single-celled organisms that can form temporary colonies, provides a window into how multicellularity might have evolved. These organisms can live perfectly well alone, but when conditions change, when food becomes scarce or predators appear, they do something remarkable. They come together. They form spheres and sheets and chains, each cell suddenly part of something larger than itself. The molecular basis of this transformation is surprisingly simple. A protein called ROS-R controls colony formation. When activated, it triggers expression of genes for adhesion proteins, allowing cells to stick together. But it also suppresses genes for cell division, ensuring the colony doesn't grow too large. It's a molecular switch between individuality and collectivity, and it can flip in minutes. Your body is a civilization. 37 trillion cells, more than all the stars in our galaxy, working in perfect coordination. Every second, millions of cells die and are replaced. Every heartbeat requires the synchronized contraction of billions of muscle cells. Every thought involves cascading electrical signals across billions of neurons. This isn't just cooperation. It's a symphony of impossible complexity, and it works so well that you never notice it happening. The scale of cellular cooperation in your body defies comprehension. Your heart beats 100,000 times per day, each beat requiring the coordinated contraction of millions of cardiac muscle cells. These cells are electrically coupled through gap junctions, protein channels that allow ions to flow directly between cells. When one cell depolarizes, the electrical signal spreads like a wave, ensuring perfect synchronization. It's been doing this since before you were born. and it will continue until your last breath. Your digestive system is another marvel of cooperation. It's not just human cells working together, but a vast ecosystem. Your gut contains 100 trillion bacteria, 10 times more than human cells in your body. These bacteria don't just hitchhike, they're essential partners. They break down complex carbohydrates your own enzymes can't touch. They synthesize vitamins K and B12. They train your immune system to distinguish friend from foe. Without them, you would starve no matter how much you ate. Dr. Michael Levin from Tufts University studies how bodies maintain this coordination. His groundbreaking research on bioelectricity reveals that cells don't just follow genetic instructions. They communicate through electrical fields, creating body-wide patterns that guide development and regeneration. When Levin's team manipulates these electrical patterns, they can cause frogs to grow eyes on their stomachs or regenerate lost limbs. The implications are staggering. Cooperation isn't just chemical, it's electrical, creating an information network that spans the entire organism. Levin's work reveals that cells can store pattern memories in their electrical networks. Planarian flatworms, when decapitated, can regenerate their heads. But if you briefly alter the electrical patterns during regeneration, they grow heads with different shapes, and amazingly, these new shapes persist through future regenerations even without further intervention. The electrical pattern has rewritten the morphological memory. Consider your immune system. It's not just a defense force, it's a molecular intelligence agency. T cells and B cells patrol your body, checking molecular passwords on every cell they encounter. Dendritic cells act as scouts, capturing invaders and presenting their molecular signatures to other immune cells. Memory cells maintain libraries of past threats, some keeping watch for decades. It's a distributed intelligence network with no central command, yet it can distinguish between trillions of different molecular patterns. The sophistication of immune recognition borders on miraculous. Your immune system can distinguish between billions of different antigens, including ones that have never existed before. It achieves this through a process called VDJ recombination, randomly shuffling gene segments to create unique antibodies. It's evolution happening in real time inside your body, with B cells competing to produce the best antibodies against each threat. The principles of cellular cooperation echo throughout the living world. From the smallest bacterial colonies to the largest animal societies, the patterns repeat: communication, specialization, sacrifice, and mutual benefit. But nowhere are these patterns more elaborate than in the social insects. Dr. Deborah Gordon from Stanford studies harvester ants in the Arizona desert. Her research reveals that ant colonies operate without central control. No ant understands the colony's overall state. Instead, they use simple rules based on chemical interactions. When an ant encounters another ant, they touch antennae, exchanging chemical information about what task they've been performing. If an ant meets many foragers, it might switch to foraging. If it meets many nest maintainers, it might switch to nest maintenance. From these simple interactions emerges complex colony behavior that can adapt to changing conditions. The parallels to cellular cooperation are striking. Just as cells use chemical signals to coordinate behavior, ants use pheromones. Just as cells specialize into different types, ants differentiate into castes. Just as cells undergo apoptosis for the good of the organism, worker ants sacrifice reproduction for the colony. The patterns of cooperation, evolved independently, converge on similar solutions. Consider the leafcutter ants studied by Dr. Cameron Curry at the University of Wisconsin. These insects have created one of nature's most complex cooperative systems. The ants don't eat the leaves they cut. They use them to farm fungus, which feeds the colony. But it goes deeper. The ants carry bacteria on their bodies that produce antibiotics to protect the fungus from parasites. It's a four-way partnership: ant, fungus, bacteria, and even viruses that infect the parasites. Each partner is essential. Remove one, and the whole system collapses. The fungus gardens of leafcutter ants are agricultural marvels. Worker ants of different sizes perform specialized tasks. The largest workers cut leaves and carry them back to the nest. Medium workers process the leaves into mulch. Tiny workers tend the fungus gardens, removing contamination and spreading fungal growth. The very smallest workers ride on the leaves being carried, protecting carriers from parasitic flies. It's division of labor refined over 50 million years of evolution. This isn't unusual, it's the norm. Dr. Margaret McFarlane-Guy from the University of Hawaii studies the bobtail squid, which harbours bioluminescent bacteria in a special organ. The bacteria provide camouflage, eliminating the squid's shadow in moonlight. The squid provides nutrients and shelter. Every morning, the squid expels 95% of the bacteria, keeping only the brightest. Its cooperation with quality control, partnership with performance reviews. The biochemistry of this partnership is exquisite. The squid's light organ has evolved tissues that focus and direct the bacterial light. The bacteria, in turn, have evolved to produce light only when they reach high density, ensuring bright illumination. The squid even has biochemical sensors that can detect the difference between its symbiotic bacteria and potential pathogens, welcoming one while rejecting the other. Humans have taken cooperation to unprecedented levels. Dr. Michael Tomasello from Duke University argues that human cooperation is qualitatively different from other species. We don't just work together, we share intentions. We create joint goals that exist nowhere in nature except human minds. When two people carry a log together, they're not just coordinating movements, they're sharing a mental representation of what they're trying to achieve. This shared intentionality appears early in human development. By 18 months, children engage in cooperative games with shared goals. They help adults complete tasks without being asked. They show concern when cooperative partners stop cooperating. These behaviours appear before language fully develops, suggesting that cooperation is foundational to human psychology. Here we are, at the edge of another transformation. Artificial intelligence is learning to collaborate with humans and each other. Synthetic biologists engineering new forms of cellular cooperation. Brain-computer interfaces creating hybrid cooperations between minds and machines. We stand where those ancient cells stood 2 billion years ago, at the threshold of a new kind of togetherness. The parallels are uncanny. Just as those first cells faced an oxygen crisis that forced them to cooperate or die, we face climate change, pandemic disease, and technological disruption that demands unprecedented global cooperation. Just as those cells had to overcome the ancient law of competition, we must transcend tribal instincts that evolved for a world that no longer exists. CRISPR gene editing allows us to rewrite the contracts between our cells. Dr. Jennifer Doudna, who won the Nobel Prize for developing CRISPR, describes it as molecular scissors that can cut and paste DNA with precision. But it's more than that. It's the ability to engineer cooperation at the molecular level. Scientists are using CRISPR to create cells that cooperate in new ways. Immune cells that team up to fight cancer, bacteria that work together to produce biofuels, plants that share nutrients more efficiently. Organoid research grows miniature brains where human neurons cooperate in dishes. Dr. Sergiu Pasca at Stanford creates assembloids, brain organoids from different brain regions that wire together and create circuits. These cellular cooperatives can model disease, test drugs, and might one day repair damaged brains. It's cooperation in a petri dish, but with profound implications for understanding consciousness itself. Xenobots, living robots made from frog cells, demonstrate programmable, biological cooperation. Created by Dr. Josh Bongard at the University of Vermont, these organisms are neither traditional robots nor typical animals. They're something entirely new. Cellular cooperatives designed by artificial intelligence and built from living tissue. They can move, heal themselves, even replicate. They represent a new form of life, one where cooperation is designed rather than evolved. We're not just observing cooperation anymore, we're designing it. Dr. Sam Bowles from the Santa Fe Institute studies how human cooperation might evolve from here. His models suggest we're at a critical juncture. Technology enables cooperation at unprecedented scales, but also creates new ways to defect. Social media connects billions, but also spreads division. Artificial intelligence could be humanity's greatest cooperative tool, or could fragment us further. The mathematics of future cooperation are complex. Network effects mean that cooperative systems can grow exponentially once they reach critical mass, but they can also collapse just as quickly if trust erodes. Dr. Robert Axelrod's research on the evolution of cooperation shows that successful cooperative strategies share key features. They're nice, never, defect first; retaliatory, punish defection; forgiving, return to cooperation after punishment; and clear, easy for others to understand. The blockchain represents one attempt to engineer trustless cooperation. Systems where cooperation doesn't require trusting other participants. Smart contracts automatically enforce agreements. Distributed ledgers make cheating impossible. It's cooperation through code rather than culture. But critics argue that removing trust from cooperation misses the point, that trust itself is valuable, that the vulnerability required for cooperation is what makes it transformative. The lessons from that first cellular merger have never been more relevant. Competition alone leads nowhere. The archaeon and bacterium that tried to destroy each other would have died in obscurity. But when they stopped trying to win against each other and started winning together, they inherited the Earth. Consider what's happening in medicine. CAR T therapy engineers immune cells to cooperate in fighting cancer. But it goes beyond individual treatments. The Human Cell Atlas project involves thousands of scientists worldwide cooperating to map every cell type in the human body. The COVID-19 vaccine development saw unprecedented cooperation between companies that usually compete. Moderna and Pfizer shared information. Oxford and AstraZeneca shared technology. It was cellular cooperation scaled up to corporate cooperation, saving millions of lives. In ecology, we're discovering that cooperation extends far beyond what we imagined. The Wood Wide Web, fungal networks that connect forest trees, allows trees to share nutrients, warn each other of insect attacks, and even support sick neighbours. Dr. Susan Simard from the University of British Columbia has shown that mother trees nurture their offspring through these networks, reducing competition to help them grow. Forests aren't collections of individual trees competing for resources. They're superorganisms cooperating at scales we're only beginning to understand. Climate change represents perhaps the ultimate cooperation challenge. It's a tragedy of the commons on a planetary scale. The atmosphere belongs to everyone and no one. Every nation benefits from others reducing emissions while wanting to emit freely themselves. Game theory suggests this should be unsolvable, yet we see glimpses of hope. The Montreal Protocol successfully eliminated ozone-depleting chemicals through global cooperation. It worked because it aligned incentives, provided clear verification mechanisms, and helped developing nations transition. It proved that planetary cooperation is possible. The Paris Agreement attempts something similar for carbon emissions, though with mixed results. The challenge isn't scientific or technological. We know how to solve climate change. The challenge is cooperation. Look at your hand. In just the skin you can see, there are more cells than there are people who have ever lived. Each cell carries mitochondria, descendants of that ancient partnership. Each mitochondrion contains DNA from a bacterium that lived 2 billion years ago. You are not an individual. You are a colony, a civilization, a walking testament to the power of cooperation. But you're more than that. You're also part of larger cooperatives. Your family sharing genes and resources, your community sharing labour and culture, your species sharing knowledge and technology. Life is cooperation all the way up and all the way down, from molecules to civilizations, from bacteria to biospheres. The neurons firing in your brain as you process these words, they're cooperating. The muscles adjusting your posture, cooperating. The immune cells patrolling your bloodstream, cooperating. Even the bacteria in your gut, breaking down your last meal, cooperating. You are cooperation incarnate, a hierarchical symphony of togetherness that stretches from molecules to consciousness. Dr. Lynn Margulis, who first proposed endosymbiotic theory, spent her final years studying Gaia theory with James Lovelock. They proposed that Earth itself might be understood as a cooperative system, with life and geology interacting to maintain habitable conditions. The oxygen in our atmosphere, the salt in our oceans, the temperature of our planet, all are regulated by feedbacks between living and non-living systems. Cooperation not just between cells, but between kingdoms of life, between life and rock, between earth and sky. Recent research supports aspects of Gaia theory. The Amazon rainforest creates its own rainfall through transpiration. Plankton in the oceans produce dimethyl sulfide, which seeds clouds that reflect sunlight and cool the planet. Weathering of rocks by bacteria and plants removes CO2 from the atmosphere, regulating temperature over geological time. Earth's habitability isn't an accident. It's maintained by countless cooperative feedbacks we're only beginning to map. We began this journey in the darkness of an ancient ocean, with two cells drifting toward each other. We've travelled through billions of years, from molecular mergers to human civilizations, from cellular contracts to global cooperation. And in all that time, across all those scales, the lesson remains the same. Alone, we are limited by what we are. Together we become what we never imagined possible. Every friendship echoes that first partnership. Every act of kindness reverberates with 2 billion-year-old wisdom. Every moment you choose connection over isolation, collaboration over competition, you're honouring an ancient contract written in the very structure of your cells. Think about the profundity of this inheritance. The love you feel for family and friends, the satisfaction of working with a team, the joy of belonging to a community. These aren't cultural constructs. They're biological imperatives encoded in every cell of your being. The pull toward togetherness that humans feel isn't weakness or sentimentality. It's the deepest wisdom of life itself, learned through billions of years of trial and error. The universe spent 10 billion years perfecting the art of being alone. Stars burned in solitude. Planets spun in isolation. Even when life emerged, it spent another billion years in single-celled solitude. But then, in one impossible moment, two cells chose each other. And in that choice, in that first successful cooperation, the universe discovered something extraordinary. It discovered that the whole really can be greater than the sum of its parts, that 1 + 1 can equal 3, or 1,000, or 37 trillion. That cooperation isn't just a strategy for survival. It's the strategy that builds complexity, creates consciousness, enables love. We started by asking when life discovered teamwork, but maybe that's the wrong question. Maybe the question isn't when life discovered teamwork, but when teamwork discovered life. Because what is life, really, but cooperation made flesh? What is consciousness but cooperation made aware? What is love but cooperation made sacred? In your cells, that ancient partnership continues. Every breath you take, every thought you think, every beat of your heart is powered by the descendants of that first merger. You don't just carry the evidence of that moment. You are the evidence. You are the proof that cooperation works, that togetherness triumphs, that the universe's greatest discovery was the simple, revolutionary idea that we're better together. The moment life discovered teamwork, it discovered the future. It discovered us. And now, as we stand at the threshold of new forms of cooperation, with artificial minds, with engineered biology, with planetary systems, we carry forward that 2 billion-year legacy. The question isn't whether we'll cooperate. Life has already answered that question. The question is how we'll cooperate, at what scale, with what purpose. The answer lies not in our technologies or institutions, but in remembering what we are. Cooperative miracles, each one of us a society of trillions working as one, each society part of something larger still. When we remember this, when we truly understand that cooperation is our deepest nature, the path forward becomes clear. We don't need to learn cooperation. We need to remember it. We need to honour it. We need to embody it at every scale, from the molecular to the global. Because in the end, the moment life discovered teamwork wasn't just a moment in the past. It's a moment that continues in every heartbeat, every breath, every gesture of connection. It's happening right now, in you, as 37 trillion cells work as one to create the miracle of consciousness contemplating its own cooperative nature. The moment life discovered teamwork, it discovered that the universe could know itself. And through you, through all of us, it continues that discovery, one cooperation at a time. If you enjoyed this journey, consider subscribing to Omni, where we explore the hidden stories of science, nature, and the universe. Every like, every comment, and every share helps these ideas reach more curious minds. Thanks for watching, and as always, stay curious.